really glad that my friends and family can be here to watch my event. And today I'm very, it's an honor of me to present you my thesis titled Genetic and Environmental Prediction of Opioid Cessation using GWAS machine learning and a mouse model. And first, we all know the opioid epidemic. From this graph, we can visualize how the opioid epidemic spread across the country measured by unintentional opioid overdose deaths in the recent 3 decades. On a molecular level, when opioid such as morphine binds to its receptor, G protein coupled receptor, the G alpha and G beta gamma subunit of the receptor will dissociate, and which leads to the increased efflux of potassium and inhibits the influx of calcium. And furthermore, the dissociation of the G protein coupled receptor will also inhibit adenylate cyclase, which will result in anergistic effect. And the opioid receptor bonded state will be desensitized and recycled back by recruitment and binding with beta-arrestin, which will lead to the tolerance and insufficient anergistic effect. But when opioid is prescribed, is used in the long term, the escalating doses of opioid and persistent activation of the receptor will increase the tolerance, which result in attenuated anergistic effect. And opioid-related research has been focused on opioid use disorder, which has a contribution from both genetic and environment. The heritability among uncorrelated individuals are 0.43 to 0.5, while the heritability among homozygotic twins can be as high as 0.7. However, currently the most effective treatment for opioid use disorder is still opioid maintenance, such as methadone treatment. However, as high as 80% of patients don't even receive the treatment, which leads to the question: what are the genetic and environmental factors associated to successful opioid cessation? Opioid cessation is especially difficult to be achieved in the long-term users. Here we measured one's opioid use disorder severities based on DSM-5, which is a popular diagnostic criteria. On the left, it is the details of the criteria, the diagnostic criteria. It listed 11 level symptom counts. We measured the most severe group. We studied the most severe group of people who have met at least 2 symptoms out of the 11 in their whole lifetime. And then we asked the question: how old were you the last time you used an opioid drug? One thing to note that this question is asked for illicit opioid use only. And with the answer more than 1 year ago, we call it they have achieved opioid cessation. With the answer less than 6 months ago, we call them that they have not achieved opioid cessation. And our study sample are derived from a large cross-sectional study, which were mainly recruited for the genetic study of opioid, cocaine, and alcohol dependence. And we have small correlated individuals in this data set as well. And this data set has both African Americans and European Americans. We start with more than 6,000 people from both population. And after throwing out the people who are not meeting the criteria, we end up with 1,192 African Americans and 2,557 European Americans who met our criteria. All of those people have been diagnosed, interviewed by a large diagnostic questionnaire which has more than 4,000 variables recording their drug use activities and the psychiatric disorder. And furthermore, a big subset of this population, we have the genotype data, which allow the genetic study of the same phenotype. And here is our hypothesis. We hypothesize that both opioid use disorder, both genes and the environment can contribute to opioid cessation, and we want to use a human model to find those factors. And in addition, since human model, especially using computational method, we tend to find lots of false positives. And we would like to use a mouse model that have display different opioid withdrawal behavior to find the genetic overlap between mice and humans. First, I would like to talk about finding the genetic factors related to opioid cessation using genome-wide association study. So what is genome-wide association study? We all know that DNA is made of single nucleotide. The common single nucleotide variation is called a SNP. And genome-wide association study is an approach that performs a genome-wide scan through a genome-wide scan with assumption that the disease-linked variant is more likely to be observed in the disease population rather than the control population. From this graph, we can see SNP3 is more likely to be linked to the disease. The sample demographics is as following, and besides the African Americans and European Americans I mentioned previously from our cross-sectional data set, we were also very fortunate to have an independent data set obtained from our collaborator, which we received the summary level statistic. And on average, our sample have severe lifetime opioid use disorder. You can see the mean opioid use disorder criteria count is more than 6. Despite their opioid use despite their opioid cessation status. So here is how we set up the genome-wide association study. We test on high-quality common SNPs, and because it's a binary trait, we use logistic regression. Because we have related people, we use generalized estimating equations to adjust for the correlation family structure, and we adjust for covariates such as age, sex. Since population structure can be a great confounder for genetic study, we adjust for population structure using principal component 1 to 5. We also adjust for batch effect using genotype by adding the genotype platform as a covariate. And then we filter out variants with low minor allele count in cases. And also we filter out the results that came from only one data set, since they are more likely to be spurious. And here is the overview of the genome-wide association study. From the top to bottom, they show the association signal from the combined population, African American population only, and European ancestry population only. On the left hand side, it's a Manhattan plot. It showed the x-axis is a chromosome location, y-axis is negative log 10 p-value. The higher the number is, the more significant it is. Red line indicate a genome-wide significant cutoff. Each dot represents a SNP. And as we can see from these 3 plots, there's no genome-wide association finding, which is not very surprising, because I have calculated for the top variant to be genome-wide association, to be genome-wide significant variant, we would need additional 300 cases to achieve 80% of power if the observed effect size is a true effect size. And on the right-hand side is a quartile plot. It shows the expected p-value distribution over the observed p-value distribution. We can see it's evenly distributed around the diagonal line until the very end, which showed that our test statistics should not be invalid and less prone to inflation. And here is the top table of the variants we have identified. The first 3 columns are the information about that specific SNP. The 4th column is indicate the genes that variant mapped is closest to, and then the association signal in African ancestry population, European ancestry population, and the combined. And several genes shown here has been discussed by others in a similar field. For example, PTPRD, protein tyrosine phosphatase receptor D, which is abundantly expressed in central nervous system, and it has been previously studied in the context of opioid reward effect. And MYOM2, this is a gene that is highly expressed in muscle tissue. And previous genome-wide association studies have identified variants within this gene are related to alcohol dependence. And using the genome-wide association study summary statistics, we find two pathways are significantly enriched for this phenotype. And all of both pathways has been indicated in opioid-related effect. And also the representative genes from this from these two pathways are indicated to be participated in opioid metabolism and opioid downstream effect. Next, since our sample are collected for a large genetic study for opioid dependence, cocaine, and alcohol dependence, we want to know if somebody who are genetically at risk for alcohol, cocaine, or opioid dependence can be more predictive of opioid cessation. And using an independent summary statistic obtained from UK Biobank, we want to study if one is more genetically susceptible to quit drinking, smoking, or having lower back pain are predictive of opioid cessation. To do that, we calculate polygenic risk score by calculating a sum total of top variants associated with these traits on the opioid cessation subjects, individuals. And here are the results stratified by population. First, we can see that the cocaine severity has a significant overlap with opioid cessation phenotype. And next, we were surprised that we failed to find any significant genetic overlap between opioid severity and opioid cessation. Next, we find that being ex-smoker, former drinker, or having lower back pain are all significant predictors of opioid cessation. So here is a summary of the genome-wide association finding. First, we identified subgenome-wide variants for opioid cessation that were mapped to biologically relevant genes of addiction traits. And those genes from opioid cessation were enriched in opioid-related pathways. And in the end, we find quitting drinking, smoking, or experiencing lower back pain can significantly predict opioid cessation. Next, I want to switch gear and talk about identify the non-genetic factors related to opioid cessation. To do that, I use machine learning with feature selection. So here I use supervised machine learning method, and the workflow is shown on the right. After the raw input, so we have a high dimensional data set. Each person, we have 4,000 variables. So traditional method, statistical method, is not good at really extracting features and doing predictions with limited sample size. That's why we use other machine learning methods. And after the raw input are pre-processed to get rid of the redundancy and normalizing the same scale, 90% of the genetic variable, 90% of the data is used as training set, and 10% was used as test set. And the supervised machine learning method is able to draw the mapping between input and output, whether or not somebody is able to quit using opioid. And after training this model several times, and until we are confident enough, we can apply this mapping function to the test set to see how well it can predict opioid cessation. And lots of literature have identified population differences in opioid use and treatment. From this graph, we can see that African Americans have a significantly lower opioid treatment completion rate compared with European Americans. And also, from the current literature, we know that opioid overdose deaths has increased more rapidly among African Americans, while the European Americans are still the leading population to have opioid use problem. So because of that, we hypothesized that there could be a different set of non-genetic factors that are contributing to one cessation behaviors among different populations. So before I talk about the different machine learning methods, I want to spend a moment to talk about the variable pre-processing step, because we know garbage in, garbage out. And so we have two populations, African Americans and European Americans. Each population we have around almost 4,000 variables to start with. After we remove the variable with the same level that has a uniform level and remove the redundant information such as the interview date, our variables can be split into binary and non-binary categories. And for the binary variables, this is a real life data set, so it's pretty messy. So there can be refusal or unknown and just missing. So after we recode all of those occasions to missing, we will throw out the variables that has more than 10% of missing, because we are less likely to confidently impute them or to guess the value of them. And for the variables that has less than 10% missing, we thought that we can learn some information from them. So that's why I code them to a different level to try to obtain as much information as possible. And the same procedure has been applied to categorical variables and ordinal variables. And for the continuous variable with missing, I use mean imputation to fill in the missing with a mean value. And I use a z-score normalization to normalize them to a mean of 0 and variance of 1, so that they are comparable in the same scale. In total, I have 15 variables from African Americans and 38 variables from European Americans to start with. So here is the content of the diagnostic questionnaire. You can see it has many sections, and a lot of sections are drug use related, and they might overlap a lot based on different individuals. So we want to know, without certain information, can we still confidently guess the opioid? Can we still confidently predict the opioid cessation outcome? That's why we came up with 3 analysis plan. The first one includes as much variables as possible. Only remove those ones who are immediately close to the outcome. The second model, we take out all the variables from the drug use section, from the opioid section. The third variables, we take out all the variables from the drug use section. And the sample demographics were very similar to the genome-wide association study. Note that people who achieved opioid cessation tends to be older and more female sex than those who have not achieved. Because we don't know what the true underlying model for our data set is, I try to use different types of methods to cover, with the hope that I can identify as much potentially associated variables as possible. So I use 2 linear methods, 2 nonlinear methods, and I'm going to talk about them in great details. The first one is lasso. Lasso is extension of ordinary least squares. Instead of minimizing the least square, instead of minimizing the squared difference between the observed and the predicted, the lasso imposed a L1 regularization term on the beta coefficient with a positive constraint, positive shrinkage, positive constraint. So with the idea is that since we're minimizing this function as a whole, some variables are forced to shrink to 0. And this method is known to be less stable since it adds more bias. And this is how I did the training. The training set were split into 10 folds with equal case control ratio. And so this is course validation. 1 fold, 9 folds are used as a training set. 1 fold is used as a validation set until each fold has been used as a training set for at least until each fold has been used as a validation set. And then we find the ideal hyperparameter in this case is lambda associated with our model that yields the highest accuracy. We apply this parameter to the test set to get the accuracy. And the next method I used is the next linear method I use is support vector machine. Support vector machine tries to find the best separating hyperplane between two classes with the hope that, not with the hope, try to find the best linear separable hyperplane between two classes with maximum margin. The idea is good features can nicely separate two classes. Therefore, they will have big margins, and compare those with those who cannot separate very well. In case of overfitting, we introduce the concept of soft margin to allow certain level of misclassification error. So the support vector machine used support vectors, the data points that were used to draw the decision boundaries to do the classification. So therefore, it is relatively more accurate. But this method is able to pick correlated variables. So similarly to lasso, I also use tenfold cross validation, and I fit the ideal parameter C learned from cross validation to the test set to get accuracy. And the first nonlinear machine learning method I used is random forest. And so random forest use sample with replacement, drawing from all the random forest builds many decision trees by using sample with replacement from all training data. The idea is that it builds many high variance, low bias decision trees, and on average they are low variance and low bias. This is an ensemble learning method, and each variable importance is measured by Gini impurity, which measures how much information is contained in each variable. And since we build many decision trees, for the entire random forest, we weighted this impurity for each individual variables by the occurrence of this variable picked by certain trees, average through all the trees that were built. But this method has high computational complexity, and it takes a longer time to finish. The next model I used is a neural network. A neural network mimics the biological neuron, where we kind of already know that the dendrites from the neuron cells are able to take the signal from nearby and carry it to the cell body, and the axon will transduce the signal downstream, and the signal that outgoing signal can be used as input for the next nearby neurons. And similarly, artificial neurons have similar points, where people have created a set of functions to take the weighted average, the sum of the weighted sum of the nearby signals, and all the informations are accumulated to the cell body. In this case, we call it a neuron or node, and we apply the activation function. It can be linear or nonlinear, so that the signal, so that afterwards the neuron knows how much signal to carry forward. But this method, although this method is very popular, it is very powerful in modeling the inter-variable relationship, but it has lower interpretability, and it has a higher number, like more hyperparameters to tune, and it's more difficult to find a local optimum. So feature selection in neural network there are several different papers talking about different methods, and I picked this method proposed by Rowe, who have indicated that this method performed very well in the feature selection from the video data. And here is how it works. Imagine that x1 to xn, there are the set of input variables, the input features that we care about, and using the law of neural network, we can calculate how much contribution does each variable have on the each node from the first hidden layer. And then we can calculate the weighted the net average contribution of each node, each input variables to a node. And then we can apply the activation function and sum them up. That will be how much each input variable's contribution to the entire hidden layer, to the entire first hidden layer. And then we can rank them and draw a curve. And we can eyeball the curve and pick a region that we think is most accurate, that has the biggest activation potential, and do cross validation on a smaller area to identify those important features. So I use two different metrics to determine the performance of my model. The first one is AUC. It plots the false positive rate over the true positive rate at any given case control split. The range of AUC is from 0 to 1, with 1 to be the best, and 0.5 means a random guess. For the other method I use is F1 score. It is a harmonic measurement of precision and recall. Precision measures when model predicts positive, how often it is correct, and recall measures how much false negative does the model have. And F1 score prevents one to focus on only the false positive or false negative. There isn't a rule of thumb for this, for which measurement to use, but since I have a relatively imbalanced data set, I have 2-to-1 case control ratio, so AUC in this case tends to be biased. That's why I use the F1 score as a rule of thumb. So here is the accuracy plot. On the left-hand side shows the accuracy plot in European Americans, and the right-hand side is African Americans. And the x-axis is stratified by different machine learning methods. Y-axis indicates the accuracy, and both methods has accuracy ranging from 0 to 1. So we can observe that like throughout in both population, AUC is consistently higher than F1 score. This is not surprising because AUC tends to perform better in the unbalanced data set. And next we can see that as the model goes from 1 to 3, the accuracy dropped. This is also not surprising because recall we have 3 models, and each model have a subset of variables that is from the previous model. So as a model, the information contained goes down, we are supposed to see that the accuracy goes down. And thirdly, we can see that as there wasn't a method that is very much outperform the rest of methods, but SVM showed that it is like marginally better than the rest of methods. But because the purpose of me using this all 4 different machine learning methods was to identify as much as important variables as possible. So after that, I summed all the important variables up and run a stepwise regression to see what are the independent predictors of opioid cessation. So I find, so here are the representative population-specific factors. So among African Americans, we observed that higher gambling severity, HIV positive, being a female are associated to the opioid cessation, while in European Americans, there are 3 others. And I can maybe pick 1 or 2 to explain why I think they are different. For example, some publications have identified that African Americans are more likely to report gambling problems rather than European Americans. That's why we see this in African Americans. Also, HIV positive is associated with higher likelihood of quitting using opioid. So we have 2 explanations for this. One is the sick quitter theory, where people are too sick to use opioid to feel high from it. The other is that HIV positive people tends to stick with the treatment really well, so they will probably like to stick with the opioid treatment very well as well, so that they can help out. Those things can help them to quit. And for the European Americans, we find that less antisocial behaviors are associated with higher likelihood of opioid cessation, since there's some other publications that have shown that for teenagers, for European American teenagers, the antisocial behavior level is associated with initiation of drug use, which might explain the reason. But so the difference we have observed could also be the other way around. If we have the same number of samples, have the same rate of the questionnaire same questionnaire completion rate. So that's a thing to be validated. Although the population-specific factors are different, we find a common cessation profile. People who quit using opioid, they tend to be older. They started using drugs later, and they use drug for a shorter amount of time, and you can read the rest. And there are implications for this study. First is that there's a need to develop treatment plans for people with opioid use disorder with other psychiatric disorders. The second is that beginning treatment earlier in the disease course might help their opioid cessation, since people who have used less time tends to be able to quit better. And also, it might be beneficial to perform drug screening on people with psychiatric disorders, since they might be less likely to report their drug use problem. The most important, in the end, most importantly, our result is our test was asymmetrical, which means we don't have any hypothesis to start with. So anything that we find might have some noise in between. So it needs future validation to become a real suggestion to be used in the clinics. Next, I want to talk about using a mouse model with different opioid withdrawal behaviors to find a genetic overlap between mouse and human. So here I use F2 recombinant mice, which was derived from two mice substring. F2 recombinant mice was first designed for fine QTL mapping, since they are less genetically complex. And previously, opioid-induced behavior traits on those F2 recombinant mice were measured, and we have observed that a marker on chromosome 1 is moderately associated to the behavior differences. So here are what has been done on these mice. We find that a subset of F2 recombinant mice, they display different drug sensation measured by locomotor activity, and different drug-induced withdrawal behaviors measured by elevated plasmase. And so these are all done by the previous PhD student. And she finds that interval on chromosome 1 might be associated with these two phenotypes. And previously, I have done the RNA-seq analysis, regressing the gene expression level for gene expression profile to the chromosome 1 marker. And I have identified around 3,000 genes to be differentially expressed. But we thought that maybe some other markers across the genome might also influence such traits. That's why we want to know how many genes are transregulated in general. And we found that 34 95 transregulated genes. And because we have identified thousands of transregulated genes, we want to narrow them down to see which genes can be are associated with certain behaviors. To do that, we use weighted gene co-expression network analysis. And here are the workflow on the right. The idea is to cluster the genes into small groups so that we can see which group of genes are significantly associated with certain behavior traits. First, we construct the gene co-expression modules based on the co-expression patterns of those genes. And then we can link those genes to these gene clusters to the mice behavior traits, such as locomotor activity and withdrawal behaviors. And then after identifying the gene clusters that are significantly associated with those behavior traits, we can find half genes, the genes with the most connectivity, to study their to be the most important genes to study. And in total, I have identified 15 gene clusters with a minimum of 60 genes, and we can see that a pink and the purple module, they all, maybe it's too small, but the pink and purple module, they all showed significant association to chromosome 1 marker and other opioid-related phenotypes. And a close-up look of those genes, genes in the pink and purple module also show a significant correlation to opioid-related behaviors. And from those two gene modules, we have identified several gene homologs that overlap with human findings from the genome-wide association study. And two of them has addiction relevance. One is HNRNPU, which was studied as a hub gene in the ethanol-exposed mice brain tissue by another investigator, and a gene in this family, HNRNPH1. Cameron is very familiar with this gene. His lab studies the influence of this gene on methamphetamine response in mice. So, and another gene is that we find to have addiction relevance is GRB1. The knockout of this gene in the parental mice, in the father mice, have been shown to have those mice make fewer impulsive choices than the mice that than the mice that with this gene. So my research has certain limitations. I want to talk about the limitations. And from the first part, finding genetic factors for opioid cessation, I used cross-sectional data. It's very likely that people who quit using opioid, they start using it again. But there are literature shows that people who have quit for a certain time, they are more likely to quit again. And we also have limited statistical power given the sample size. We have not identified any genome-wide significant finding because of probably largely due to the power issue. And also the genetic overlap between opioid cessation and other addiction traits that I showed earlier was derived from like same big sample pool, which may cause the overestimation of the opioid of the genetic overlap. And for the machine learning part that I have talked about, we hope to have more detailed information about the treatment and also the reason of their first use. Those detailed information was not collected by the questionnaire. And we observed the opioid cessation differences. Those differences could be due to the response rate, the questionnaire response rate, and also the machine learning. We have small families from our data set. A lot of machine learning methods do not fully adjust for family correlations. I don't know how to. I have read a lot of papers. I have not found a good way to solve that. And for the mouse study, we know that mouse is a good proxy for human because it has more than 99 gene homologs compared with human beings. But lots of factors related to opioid cessation in mice, in human can still not be modeled very well in mice because they don't have other social pressure to quit. And we also do not have the same level of transcriptomic data from human brain to do the same level of comparison. So in conclusion, we've identified novel genetic variants and biological pathways that are potentially regulating opioid cessation through a genome-wide association study. We also identified phenotypic factors that are contributing to opioid cessation through machine learning feature selection. And we have identified two important gene homologs with opioid addiction relevant traits to human. And this framework can be applied to other disease fields where both phenotypic and genotype data are available. In the end, I want to thank everyone in my community to help me in all of those years. First, I would like to thank my PI, Dr. Lindsay Farrer, to believe me ever since I was a student. And before I come to this lab, I have no skills related to anything I have just talked about. And he believed in me. He believed in me and accepted me as a PhD student and encouraged me, invested in me, and also Dr. Farrer is very well connected in the research field, and I got a chance to collaborate with lots of different collaborators and got lots of good feedback and comments from them. And secondly, I want to thank Rick for always helping me with no matter my research questions, if I ask you the research questions or it's just English problem, you are always very patient and very helpful, give great suggestions all the time. And I also want to thank Dr. Richard Thesis to make my research so clinically impactful. We sit down a lot and talk about how do we make the clinicians understand our research. And also I want to thank Cassie for always helping me understand statistics better and teach me all the great classes. I attend almost all the classes that you taught.
Did she pass?
And also I want to thank Cameron for being very helpful on the mouse project and giving me opportunities to get involved in lots of other projects outside of my PhD project and to practice thinking in a very data science way. I also want to thank Dr. Mark Cohn for always answer questions I have with machine learning problems and willing to sit down with me to go over my machine learning thoughts. And lastly, I also want to thank Shumita for having such a great program and get me connected to the past students and organize such a great program with lots of fun student activities and recommend me with different resources, career-wise and academic-wise. I also want to thank the past and current members. First, I would like to thank Yi Ma, who was a past member as a postdoc. She helped me. She introduced me to this lab, and also Jiayi and Xiaoling for letting me be more proficient on computational side of getting me familiar with doing work in a computational way. And then I want to thank Cong for being very helpful in the first 2 years. I didn't know much. And I also want to thank John, Bianca, and other PhD students in my lab for always giving me good suggestions and just being there for to help me. And I also want to thank our collaborators for giving such great comments, being so helpful for the for different revisions of the paper, and the past and current student who helped me with the RNA-seq projects. I will not know how to do that if they don't help me. And I also want to thank the PIPS program in biomedical sciences that recruited me to be a PhD student at first to start this wonderful journey, and also program in genetics and genomics. We have fun student activities and we have lots of great talks. And finally, I want to thank BU Best, Amanda specifically. She sat down and go over my slides with me several times to make sure that I have a good presentation skills. And also, I want to thank my husband, RJ. When we first met, I wasn't a PhD student. I was still a master student, and I won't even know if I will still stay in Boston or not. And we still hung out a lot, and I'm glad that things work out. And then it's my parents. They are here today. They flew all the way here from China, and they're still working. They have to take a week off to be here and have jet lag. Very supportive and always very loving. And I also want to thank my parents-in-law who might be watching from there. They are very loving and encouraging, and they are very motivational too. They just help me get more used to the life in the US. And in the end, I want to thank all my families and friends who helped me over the years. And I would like to take any questions if you have. And the post-defense celebration will be at 3 p.m. in room 201.